This program is brought to you by Emory University.
Hello, my name is Khalid Salita. I'm an assistant professor of chemistry at Emory University. A large number of students in the lab currently are working on trying to answer this big question, which is how do mechanical forces at interfaces, how do those forces control signaling, control fates of cells, control biochemical outcomes? That whole project evolved because of a question I had in a seminar that I was giving in the School of Medicine. So at that time, we were interested in how receptors laterally move in a membrane. But as I gave this talk and presented some data, one of the students in the School of Medicine asked if we were studying how receptors move in the direction perpendicular to the cell membrane. I said, no, we don't generally look at that. We don't have the tools to look at that. And she said that would be a very interesting thing to look at because it's known that many of these receptors are translocated to the inside of the cell. So I went back after the seminar and did literature search, and it seemed that there's very few tools to trying to measure displacement perpendicular to the cell membrane. And consequently, we started developing ways to try to measure those Z displacements. And as a direct consequence of measuring Z displacement, we decided and we realized that we can measure force. Because if we know a spring constant and you know a displacement, you then can back calculate tension. We are pioneers in this area. We're the only lab, for example, we developed the only method that one can use to image piconewton forces at the cell surface. And so it turns out that at the level of individual molecules, piconewton 10 to the negative 12 newtons is the range of force that's relevant. Those are the range of forces responsible for switching conformations of proteins, exposing cryptic sites, leading to binding events that would otherwise not occur.
The preceding program is copyrighted by Emory University.